In the last lesson, we learnt about an important and interesting family of ion channels, the voltage-gated ion channels. For a voltage-gated ion channel, the open probability depends upon the membrane potential. Furthermore, we can see that there's a complicated situation where the opening and closing of the voltage-gated ion channel changes membrane potential, and that change in membrane potential in turn affects the open probability. And so we can envisage complex interactions between the voltage-gated ion channel function and membrane potential. The dynamics of those interactions depend upon the channel kinetics, how rapidly it is changed in its open probability by the membrane potential. The voltage gating of the ion channel depends upon changes to the 3D conformation, the structure of the protein ion channel itself, and this does not occur instantaneously. The opening and closing of the ion channel, the individual opening and closing, they occur on the microsecond timescale. The voltage gating, the change in the open probability of the ion channel, occurs on slightly longer timescales, and it's those dynamics that we're going to study in today's lesson. We're going to focus again on the voltage-gated sodium and potassium conductances that we began to study in the last lesson. These, as we remember, are largely closed at resting membrane potentials of around minus 70 millivolts. They only become activated as we depolarize. As we go beyond minus 40 millivolts and above, the open probability of the voltage-gated sodium-potassium conductances increases dramatically, causing an inward sodium current and an outward potassium conductance. In order to understand how they dynamically interact on the cell membrane in real cells, we need to know about the gating kinetics. If we rapidly change the membrane potential, what happens to the sodium current in time? For the voltage-gated sodium conductance, we can imagine a situation where we have a patch clamp electrode sitting on a piece of membrane. We suck the bit of membrane into the patch electrode, and in this patch would be voltage-gated sodium channels. For a given membrane potential, we can then, using the patch clamp amplifiers, change that membrane potential, and we can then activate these voltage-gated sodium conductances. We know that the current voltage relationship for a sodium conductance is something like this. The reversal potential here at around plus 60 millivolts, activating somewhere around minus 40 millivolts, and being closed basically at hyperpolarized potentials relative to this. So let's take a situation where we start at minus 70 millivolts. At some given time, we jump the membrane potential to 0 millivolts, and we're going to be studying the single channel currents flowing through an individual ion channel inside the patch electrode. So, as we go from minus 70 millivolts to 0 millivolts, initially the open probability is very low. The ion channel is closed at minus 70 millivolts. We then jump to 0 millivolts. Now we have an inward driving force, so we're expecting an inward current to happen. Initially, nothing happens, but then after a short delay, this ion channel opens, stays open for a brief period of time, closes again, and then remains closed for the duration of the depolarization to 0 millivolts. On the next trial, when we repeat this, again we're at minus 70 millivolts, no current flow, we depolarize to 0 millivolts, short delay, ion channel opens, stays open, closes, and then again remains closed for the duration. In this case here, the ion channel is open for a slightly longer duration here with some stochastic opening and closings. And you can imagine repeating this over and over again for hundreds of trials, or alternatively, summating that current across a whole cell membrane and doing just a single voltage step and seeing the summated average currents across the sodium channels. And either way, we get the same picture. We make the voltage step from minus 70 to 0 millivolts. After a brief delay, inward current is activated. This takes maybe some 10 microseconds or so, and then the sodium conductance remains high for a period of about 200 microseconds, and then it closes. The voltage-gated sodium channel inactivates rapidly on that timescale of hundreds of microseconds. Fast, brief, transient voltage-gated sodium conductance is activated by a prolonged step to 0 millivolts. For the voltage-gated potassium conductance, things are rather different. Again, we envisage a situation where we have a glass patch electrode sucking a bit of membrane inside it, and inside this membrane we now have a potassium conductance. We can choose the membrane potential through the patch clamp amplifier, and again we take a situation where we start at minus 70 millivolts, the ion channel is closed, we have our current voltage relationships that are characteristic, which are closed until somewhere around minus 40 millivolts. We jump to 0 millivolts, we have an outward driving force, and we now study what happens at the level of single channels as we make that jump in voltage from minus 70 to 0 millivolts. And here we're looking at the single channel current through our patch electrode. We make the voltage jump, and initially nothing happens. There's a long delay, and then at some time the channel opens, stays open, briefly closes, opens, briefly closes, opens again, and this ion channel will remain open as long as we keep the plasma membrane depolarized to 0 millivolts. In another trial, we do the same. We jump from minus 70 to 0 millivolts. There's a delay. The ion channel opens, closes, opens, and stays open for as long as we care to keep the patch of membrane depolarized. We can again summate this across multiple ion channels that are present in the cell membrane, or we can repeat at the level of individual ion channels and get the mean current flow through the potassium conductance, and either way we get the same picture. We jump from minus 70 millivolts to 0 millivolts. There's a delay. Nothing happens for a few hundred microseconds, so about 200 microseconds. Nothing happens, and then the open probability of the voltage-gated potassium conductance increases, and we see an increase in the current flow through the voltage-gated potassium channels. We can directly compare the kinetics of the voltage-gated sodium and potassium currents by putting them on the same graph. And so again, we imagine going from our hyperpolarized to our depolarized state that activates both the voltage-gated sodium and the potassium conductances. They're both activated somewhere positive to minus 40 millivolts. But the major difference is in the time course. The voltage-gated sodium conductance is fast and transient. The voltage-gated potassium conductance is delayed and long-lasting without inactivation. The transient sodium conductance lasts something like 200 microseconds, and the potassium conductance is delayed by that same order of magnitude, some hundreds of microseconds, delay the activation of the potassium conductance that then remains active and doesn't inactivate, whereas here the sodium conductance has fully inactivated after just a few hundred microseconds of the depolarization. However, the voltage-gated sodium channel doesn't remain inactivated forever. We can take that membrane potential, hyperpolarize it again, say to minus 70 millivolts, and under those circumstances, the sodium channel de-inactivates, and we can go through another round of this, where we can make a step activation, and we'll again see the transient voltage-gated sodium conductance. The de-inactivation doesn't occur instantaneously, but again takes some time. We can study the recovery from inactivation by doing two-pulse experiments. And so as before, we go from our hyperpolarized to our depolarized state. We see the transient voltage-gated sodium current that becomes activated by this depolarizing step. And then at some later time, after we've repolarized again back to minus 70 millivolts, we can then study how long it takes before we recover our full sodium current. If we wait half a millisecond, we get some recovery, but not much. If we wait 1 millisecond, the recovery is already better, and after about 5 milliseconds, we're almost back to the full voltage-gated sodium current that we had from control conditions. And so we can plot an exponential recovery time course for the voltage-gated sodium conductance if we think of this as the second peak and this as the control first peak, we can look at the ratio of the second activated voltage-gated sodium conductance compared to the first one, and we find that there's a recovery time constant with an exponential form here, and the exponential for the recovery from inactivation is there on the several millisecond timescale. So we've seen some important features of the kinetics of the activation and inactivation of the voltage-gated sodium and potassium conductances. Both voltage-gated sodium and potassium conductances are inactive at resting membrane potential around minus 70 millivolt. Above minus 40 millivolt, the open probability for both sodium and potassium conductances increases dramatically, but the sodium conductance is rapidly activated in the time domain, lasting a few hundred microseconds, is then inactivated, whereas the potassium conductance is delayed. It takes some hundreds of microseconds before it even opens, and then it doesn't inactivate, but remains open for as long as the membrane is depolarized. Now, this is true in general of the voltage-gated sodium and potassium conductances that one sees, but there's also some diversity as one goes across different species and across different cells in the same species. And we'll have a little look at the basis of that in the next slides. The voltage-gated sodium channels are encoded by a family of 9 genes that are given the nomenclature NAV1.1 through to NAV1.9. All of them encode the voltage-gated sodium channel in a single gene. So it's one long string of amino acids that has transmembrane domains, and it has 4 subunits that are all part of the same string of amino acids. So this would be the N-terminal, the C-terminal, and here's subunit 1, 2, 3, and 4. And these are then the transmembrane regions that will ultimately sit in the plasma membrane and will then form the 4 subunits of the ion channel, the center of which forms the ion-conducting channel pore. Associated with the so-called alpha subunit of the voltage-gated sodium channel is a much smaller beta subunit that sits somewhere tightly associated to the ion channel proteins encoded by the alpha subunit. Together, this provides some diversity. There are 9 different sodium channel proteins, there are 4 different beta subunits, and they can then encode slightly different functions in terms of the single channel conductance and the detailed kinetics and the voltage activation. Another very important aspect that's different between the different isoforms is the cell types in which they're expressed and also the subcellular localization of the voltage-gated sodium channel. As we will learn in the next lesson, the density of the voltage-gated sodium channel is regulated very precisely, and some small subregions of neurons have very high densities of voltage-gated sodium channels, and that makes a big difference to the functioning of neurons. There's also some diversity in terms of the time course of the voltage-gated sodium channel, and in particular, some subtypes of voltage-gated sodium channels have much less inactivation than what we've discussed. Potassium channels are an even more diverse family of ion channels. There's something like 80 different genes in the mammalian genome that encode potassium channels. Not all of the potassium channels are voltage-gated. They are an important family of the potassium channels, and they're certainly the ones that are most interesting for us in terms of thinking about the complex dynamics of membrane potential, but they're not the only ones. There are also potassium channels that are gated by the intracellular calcium concentration, calcium-activated potassium channels that are so-called BK and SK channels. And there's another family of proteins, of potassium channels that are so-called G-protein coupled ion channels. These are the so-called GERK channels. And there are the tandem pore potassium channels that are involved in setting the resting membrane potential. The voltage-gated potassium channels in themselves have extensive diversity. Some of the voltage-gated potassium channels have some degree of inactivation, but on a much longer timescale than what we saw for the voltage-gated sodium channels. The potassium channels are also different in the sense that they encode individual subunits rather than the whole potassium channel in itself. And so one gene will encode the 6 transmembrane regions of the potassium channel, but it'll only encode one subunit of that. And so one gene will encode one subunit, and another gene might mix together with that and form other parts of the subunit of the potassium channel. And so you can generate even more diversity by mixing different proteins together to form the voltage-gated potassium channels. And the diversity in the voltage-gated potassium channels causes a great deal of diversity in terms of how neuronal membrane potential changes in different cell types and in different compartments of the same cell. So now we've learnt a bit about the voltage activation of the sodium and the potassium conductances. We have the voltage activation of the sodium conductance, and we have the voltage activation of the potassium conductance, both of which are activating somewhere positive to minus 40 millivolts and closed at resting membrane potentials. We also know something about the dynamics of the opening of the voltage-gated sodium conductance that opens rapidly and then inactivates, whereas for the potassium conductance we have a slower rise that takes some hundreds of microseconds. We can then begin to think about what happens inside a cell that might express both the sodium and the potassium conductances. And if we think now about the different time courses of these, you can imagine that if we take the membrane potential a little bit depolarized and we get into this regime where the sodium channel begins to activate, we could then get this brief activity of the voltage-gated sodium conductance that will drive a depolarization through the inward current through the sodium conductance. This will then inactivate, and at the same time, while the sodium conductance inactivates, the potassium conductance is activating, driving a hyperpolarization. And we'll see it's exactly this dynamic interaction between the sodium and the potassium conductances giving rise to brief transient excursions in the membrane potential with time that drives the so-called action potential. And that action potential is what we'll be studying in the next lesson. So to summarize this lesson, we've learned some interesting details about the kinetics of activation and inactivation of voltage-gated sodium and potassium channels. The voltage-gated sodium channel activates rapidly, then inactivates rapidly, and it remains inactivated until the membrane potential hyperpolarizes again, and then after a few milliseconds, it's de-inactivated and ready for the next voltage pulse. The voltage-gated potassium channels, on the other hand, are slowly activated by depolarization. It takes them a few hundred microseconds before their open probability increases, but then they remain open for as long as the membrane potential is depolarized. The complex interplay between time, space, and membrane potential and the opening of these voltage-gated ion channels causes very interesting and complex dynamics in membrane potential, and ultimately underlie the action potential signal, which is the all or none signal that we'll go on to study in the next lesson.